Cluster analysis. Cluster analysis also is a form of exploratory data analysis. And this is a technique used to classify different objects, different units of data collection into different groups based on sharing of common characteristics. In other words, you can say that this is a technique of classification of units based on similarity of observations, so that observations within such groups are similar in the case of the variable under study, and observations between the groups show higher variation. Each such group can be called a cluster. You can subdivide each cluster into subclusters, and this process can be repeated based on the analysis of more number of variables. Cluster analysis has applications in different conditions. The most important application of cluster analysis is in classification. Perhaps it is the most widely used in biological classification. Use of cluster analysis in biology started as a very effective tool to classify different types of organisms into different categories like classes, orders, families, genera and species, thus giving birth to the science of numerical taxonomy. Now it is widely used in systems of molecular classification, biochemical classification, etc. also. It has found application in molecular systematics and in the studies of phylogenies of different organisms. Now you can see certain examples of cluster analysis. Cluster analysis based on morphological, biochemical or molecular characters make it possible to classify organisms. More similar organisms show differences to a very low extent, and they occupy very close position on a tree constructed using the similarities and differences. Related organisms like apes and man occupy a closer position, while other mammals and men occupy comparatively distant positions. In the field of medicine, you can cluster diseases so that common diseases can be addressed with common protocols of treatment. In the case of history, archaeology, etc., you can find out the taxonomies of stone tools and funeral objects based on their similarities and differences. You can classify a mountain of data into several meaningful subsets using cluster analysis. And one important step in cluster analysis is standardization of data. You get different data in different units and different dimensions. And if you are not standardizing data, the effect of larger data will be higher when compared to smaller parameters. So standardization of data is very important in cluster analysis, and usually before proceeding to cluster analysis, the data are standardized so that they have a mean of 0 and a standard deviation of 1. If the variables are not converted into comparable magnitude in such a way, the analysis becomes meaningless. Usually the distances of the mean from the different values of x in the case of each variable are converted into comparable magnitudes. If the variables are qualitative in nature, they are denoted based on their presence or absence, or are given weights based on their different forms of expression. So now you can see that cluster analysis is a technique of classifying the objects of data collection, or in some cases even classifying the variables themselves into different clusters, and there are basically different methods of cluster analysis, and primarily there are 3 methods used. And the first method is hierarchical cluster, and here it creates a series of models with cluster solutions from 1 to n. that is, from all cases in one cluster to each case in an individual cluster. This is the most common method. And the second one is k-means cluster, and this is a method used to cluster large data sets. In this case, the number of clusters is assumed by the researcher prior to carrying out the analysis. And the third type is two-step cluster, and this is a method combining the k-means clustering and hierarchical clustering. K-means clustering is carried out first to identify groupings, and then hierarchical clustering is performed. This is also used in the case of larger data which are difficult to handle. Hierarchical cluster can also be called tree clustering. It is called tree clustering because by clustering, trees are developed based on the increase in dissimilarity or distance between the objects in this case. Tree clustering will successively link together objects of increasing dissimilarity. Each leaf of the tree corresponds to one object. The branching of the tree reflects the relationship between objects in terms of distance or proximity. You have to use appropriate distance measures in correlation analysis. And there are different ways to compute the distances. Calculation of Euclidean distance, Manhattan distance, Jaccard index or Jaccard similarity coefficient, etc. are adopted. Euclidean distance is the ordinary distance between the 2 points that can be measured using a ruler. It is the straight line between the 2 points. Manhattan distance is the distance between 2 points measured in right angles. Jaccard similarity coefficient measures the similarity between sample sets. Percent disagreements are also used as measures of distance in cluster analysis. And since in cluster analysis you have to link the variables or link the objects based on the affinities between them, and you have to apply certain amalgamation rules. And in the case of cluster analysis, different types of linkage rules have been proposed for the amalgamation of the objects under study. The most important ones include single linkage, complete linkage, unweighted pair group average or UPGMA, weighted pair group average or WPGMA, percent disagreement, etc. are some of the most important methods of amalgamation proposed. Single linkage is the nearest neighbor rule. Here, as we proceed from larger clusters to clusters of less and less similar objects, the distance between any two clusters is determined by the closest objects in those two clusters. Then, by cluster analysis, you will get a diagram which is usually known as a dendrogram or a T diagram. Dendrogram is the diagrammatic representation of the distance between the different objects under study. Usually it is constructed by the amalgamation of the distances between the different objects used in cluster analysis. You can call it a tree diagram also. Each leaf of the tree corresponds to one object, and the branching of the tree reflects the relationship between objects in terms of distance or proximity. Now we can move to an example of cluster analysis. Here, cluster analysis is carried out in the case of 25 genotypes of peperomia based on 15 variables. Mean values of different variables are given to you in the case of 25 genotypes, and based on their behavior, you are asked to classify them into different clusters. And when you carry out cluster analysis, since it is a little sophisticated pattern of analysis involved, we usually nowadays use statistical packages for the purpose. And here we are classifying the 25 genotypes into different clusters based on the 15 variables being analyzed. The classification will be based on the difference in the case of each and every variable, and here the pattern of analysis used is unweighted pair group average method, and we are using percent disagreement as the measure of distance. Based on percent disagreement, we are classifying 25 genotypes of data into different clusters. Based on 15 variables, we are classifying the 25 genotypes of peperomia into different clusters by cluster analysis. And when you classify the data and carry out analysis, cluster analysis using percent disagreement, you will be getting a dendrogram or a clustering diagram. A tree diagram for the 25 genotypes under study is presented here, and it is the result of cluster analysis in 25 genotypes. Using UPGMA method, so using UPGMA method, you are classifying the data and you are carrying out the analysis. First, you are getting a table showing percent disagreement. The distance between different objects, different genotypes under study based on the 15 characters will be computed, and it would be given in the form of a spreadsheet in which the percent disagreement between the genotypes 1 to 25 will be given. And from this percent disagreement, the program constructs a dendrogram, and through the analysis you will be getting a dendrogram, and in the dendrogram you can see that the genotypes have been presented at different positions in the dendrogram or the tree diagram, or each genotype represents a leaf of the dendrogram, and there are some genotypes very close and others are little away, and this type of a dendrogram has been constructed based on the percent disagreement, the percentage of disagreement between the different genotypes under study based on the 15 variables. And so a dendrogram has been produced, a dendrogram has been made, and it is the tree diagram of 25 genotypes made using unweighted pair group average method with percent disagreement as the measure of distance between the genotypes. You can see in the dendrogram that all the 25 genotypes have been presented at different positions. They have got distributed to different positions in the dendrogram based on their similarities and dissimilarities with other genotypes. When you analyze the dendrogram, you can see that the tree has been split into two at a linkage distance of 1, or the first step of branching of the tree is at a linkage distance of 1.00, and there the tree has got branched into 2 branches, cluster, or a distinct cluster which is different from all the others, and all the other genotypes stand together at this stage of linkage distance as calculated using percent disagreement. And there are only 2 genotypes in one cluster, and all the 23 genotypes are in the other large cluster. And then gradually the large cluster gets divided into several subclusters. And in the first stage, it gets divided into 2 major subclusters. Towards the upper side of the diagram, you can get the first major subcluster, which contains only 2 genotypes, the genotype 1 and the genotype 4. And then comes the next major subcluster, and it contains the genotype 5, 7, genotype 7, genotype 16, genotype 14. And then there is another very large subcluster, and it contains several genotypes, all the genotypes other than these ones. So you can see that the subclusters described earlier contain a few number of genotypes, and all the other genotypes of the second cluster are then clustered together. And you can see that, you can see it in the diagram, starting from the genotype number 2 to the genotype number 20, they are positioned differently. And then also there are different levels of splitting. In the case of the first division, in which 2 subclusters were produced, or 2 clusters were produced, and one cluster consisted of only 2 genotypes, and all the other 23 genotypes clustered together. The 2 genotypes of the small cluster again got subclustered into 2, the 21 and 11, and they formed the leaves of the tree directly without much branching of the branches. And when you go to analyze all the different subclusters of the major cluster, in the case of the first subcluster, which consisted of genotype 1 and genotype 4, the branching occurred rather earlier. You can say that it occurred between 0.92 and 0.94, between the linkage distance 0.92 and 0.94, and in the first subcluster formed from that major subcluster, 3 genotypes bifurcated, and one more genotype bifurcated directly from the subcluster at a higher level. You can say at a linkage distance of 0.98. Similarly, all the other genotypes also got positioned as different leaves of the dendrogram. And finally, if you go below, the linkage distance gets reduced, and from this diagram you can see that the genotypes 12 and 18 are very close to each other. Similarly, the genotypes 13, 15 and 17 are very close to each other. Thus, if you are analyzing the dendrogram systematically, you can find out the closest genotypes, and here the closest genotypes are 12 and 18, they are very close. It means that the variables which were taken into consideration at the time of cluster analysis showed very little difference in the case of these two genotypes. Similar is the case of the three genotypes 13, 15 and 17, and there also they might have shown very little difference between them, so that in the tree they are clustered as very nearly positioned leaves. Then when you move back, you can see that genotypes are getting split up. Several branches of the tree develop almost simultaneously, and most of the branches bear only one leaf, and some of them bear more than one leaves. At the position somewhere in between 0.92 and 0.94, there is such a very considerable level of branching, indicating a very significant diversification of the genotypes based on these characters at this level. And again, in the case of some subclusters, there are still more branches formed, and some branches are further unbranched, which end in a single leaf, and some are branched, which carry a number of leaves. So that is about a dendrogram in which the diversification or the genotype level classification of 25 genotypes of the population under study got presented, and based at different linkage levels, the branches got divided into sub branches, and finally all the branches ended in leaves, and each leaf represented a genotypes. Some genotypes were very close and some were very distant. Interestingly, the most distant genotypes in this case are genotype 11 and genotype 21, and of course genotype 1 and genotype 4 also, although they belong to the second major cluster, at a very early level, they are again getting split into subclusters and finally ending in two genotypes, genotype 1 and genotype 4. Like that, using a dendrogram, you can comment on the nature of variation shown by different genotypes based on morphological characters, based on characters obtained from biochemical or phytochemical analysis, characters analyzed using molecular tools, etc. Nowadays, the use of clustering or cluster analysis for studying the molecular diversification of genotypes of plants, animals, microorganisms, etc. Such studies are very important, very frequent and very relevant nowadays, and several such studies are being carried out regularly, which bring about exciting outcomes. So, cluster analysis is a tool that can very efficiently be used in biology. It does not mean that it has no position in other branches of science and social sciences anywhere. Based on affinities of different types, objects of data collection, units of data collection can be classified very easily based on clustering analysis. And as mentioned previously, we know that a hierarchical clustering is usually carried out, in which the number of clusters will be decided at random, and if the data are very large, or if the data set is very large, you go for k-means clustering or two-way clustering. And there are different types of calculating linkage distances and different amalgamation rules, and based on the nature of the data, you will be selecting an appropriate linkage rule and an appropriate amalgamation schedule. In the case of quantitative data, you can do it rather easily, but in the case of qualitative data, you will have to convert the data into quantitative terms. For example, if you are classifying plant species based on qualitative characters like presence or absence of stipules, presence or absence of spines or thorns, presence or absence of a particular flower color, or you can classify them based on flower colors, red flowered, white flowered, like that. And in such cases, you will have to consider the presence as 1 and the absence as 0. In molecular analysis also, very often you are getting different bands when you analyze the molecular data of the plants, animals, or human beings, and based on the molecular data, you will be deciding whether a particular locus is present or not, and presence will be marked as 1 and absence will be marked as 0. Presence of a band will be usually marked as 1 and absence will be marked as 0, and there also a similarity matrix will be developed, and from the similarity matrix the percentage of the similarity will be calculated, and based on the data obtained you can construct a dendrogram. The distances and proximities between different genotypes can be found out, and disputes on phylogeny can be solved in this way. So, cluster analysis is a very important pattern of analysis, and there are several uses. And of course, you can classify diseases, you can cluster diseases, you can classify diseases into different clusters, and you can develop treatment protocols common to each and every cluster. Thank you very much.